Thanks, Paul, for a wonderful talk. Great first session, hearing from really two of the probably biggest tool builders in the field for all of us structural biologists. It was really fantastic. Yeah, so my name is Steve Reichel, over here at PNCC. I just want to again say thanks on behalf of all of us at the center for joining us today, everyone who is in attendance and all of the speakers. I also want to make sure that we got a special acknowledgement to Omar, who really spearheaded the organization of this workshop, really did a fantastic job of putting together a marquee list of speakers. And by attendance, there's a lot of enthusiasm for all the work that's going on in this field. So my pleasure to introduce the speakers for this next session, which is kind of really going to highlight what I think are some exciting advances that AI is bringing to cryoEM data analysis, as well as some things that users should really be careful of when they're starting to work with these emerging technologies. And I think that hints at kind of the scope of what we're going to hear from our first speaker today, Mark Herzik, who's at UC San Diego. The title of his talk being Machine Learning Approaches to cryoEM Density Modification Differentially Affect Biomacromolecule and Ligand Density Quality. With that, Mark, thanks for joining us today. Look forward to your talk.
Absolutely. It's a mouthful. The title is a mouthful for sure.
I'm glad I got through
All right, let me make sure I am okay. Everyone can see? Perfect. Okay. So we're going to change gears a little bit because now what I want to focus on is really this idea of getting to the end stages of your cryoEM project and really focusing on, you have your map and what can you really do with it.
it.
So,
Mark, I'm
Mark, if
sorry to
I
interrupt.
can,
Yeah, I think we're seeing like the preview of your slides as opposed to a show view.
Is that
There you
better?
go. Perfect. Thanks.
All right. I don't have anything hidden in the slide presenter view, so I don't have to worry about it too much. Right. So the workflow for cryoEM is something that we like to distill down into this very linear workflow. It's very simplistic, ranging from, all the way from looking at your raw micrographs, 2D classification. Some people skip this for some reason, but in general, moving then into 3D. And I would say that this is by far one of the most subjective decision-making processes you have. There's tons of hyperparameters that go into this. There's tons of workshops that are oriented around how to best approach this. And I would say at the end of the day, once you have your final cryo-EM map or maps, if you have buyer's remorse, you don't want to start over in this entire process. And rather, you want to say, okay, I have my half maps, I have my final EM density. What can I do to improve the map? What can I do to interpret it the best I can? And what can I do to build the best atomic models using the software packages that we just were given privy to? And so one of the things that I think we can all agree on, both as crystallographers and as electron microscopists, is that this idea that there's a huge wealth of information that comes as you increase the resolution content of your data. And so here's a general slide that I like to show to give you an idea of what happens as you progress from, low 4 angstrom resolution where you can basically make out an alpha helix, and as you move into the three angstrom range, you really start to see, atomistic details looking at side chains, rotamers, backbone conformations. And now as we approach the 2 angstrom realm more frequently, you can really start to build in additional, ligands, waters, metals, and alternate side chain conformers. And so there's a wealth of information that you can glean as you move into increasing resolutions, which can come from both data quality as well as how you handle the maps at the end of the day. And so always I try to emphasize is no matter how good we are with our data and our processing, you at the end of the day still need to know your amino acids because depending on the microscope configuration, depending on the software you use, depending on the resolution of your final maps, the sidechain information can be somewhat ambiguous. And unless you have additional information based on your atomic model, the sequence, local chemical environment, de novo building can still be a bit misleading if the density is not of the highest quality. And so I think we've seen a couple of highlights of this so far today, but in this new age of structural biology, we're constantly being asked of, what can we do to make this more expeditious? What can we do to improve things? And importantly, how can we use machine learning to improve aspects of the cryo-EM workflow? And I would say everywhere is the short answer to this because, as you can see, just by basically going into PubMed and looking up machine learning cryo-EM, we're kind of at an exponential increase in the more recent years of what machine learning is doing for cryo-EM, ranging all the way from particle picking tools, even data collection, sample screening, all the way down to at the end stages where you're looking at map modifications. And that's more or less where I really want to focus on, today in this workshop. And so one of the things that I do want to highlight though is because this technique, this cryo-EM vitrification process, it serves as a fixative, but it's not perfect. And what I mean by that is that it does preserve compositional and conformational heterogeneity. And so that means at the end of the day, if you have a heterogeneous map, you're not going to be able to make it perfectly homogeneous no matter what software package you use. And so, because we're averaging typically thousands or even millions of these noisy 2D particle images into our 3D map or our maps, this imperfect process leads to imperfect analyses downstream that require validation. And so, one of the things that we have to be careful with in the community is that because the cryo-EM application spans everything from proteins, nucleic acids, membrane proteins, complexes, and pretty much everything in between, there is very rarely a single software package that will work with every sample, every situation without any caveats. And so, because of this, there are potential issues depending on your applicability. And so there are also assumptions in almost every software package that have to be taken into consideration. And so knowing this, yes, you can absolutely apply pretty much any and every software to your data as you see fit. But at the end of the day, you have to be careful knowing the limitations of the software and what you can and can't do with it. And so when we think about cryo-EM data, it's very different than crystallography because at the end we get an EM map. The EM map itself has excellent phases, as we've heard. Amplitudes sometimes aren't all that great, but the map is pretty invariant. And so what that means is once we generate our final map, as you can see here in this unsharpened 2.9 angstrom map, you can see decent EM density for this heme cofactor, the propionates, and these histidine ligands. This is from hemoglobin. What happens is from this FSC weighted unsharpened map, it's sometimes blurry, not as featureful as we would want, and that's due to the contrast transfer and the imaging artifacts that arise from this technique. And so what people almost always do right out the gate is they use a sharpening approach to basically improve the visibility of the backbone atoms and side chain information. And so as you can see here, this exact same density that's now been sharpened with a global B factor of negative about 120 angstrom squared. You can see in comparison that the propionate density gets better, the vinyl groups on the heme cofactor get better, and overall side chain densities improve just by applying this single map modifier. And so with this, the reason we approach these map modifiers is to do exactly what we've heard about early in today's workshop is it aids model building. And by doing so, it improves the density, allows us to interpret it a little bit better, and by improving the information content, it allows for our atomic models to hopefully refine and be more chemically correct. Now, one thing to really note here is that map modifications can either clarify or distort your map. Both outcomes are possible, and frequently, because our EM maps are heterogeneous, a single map will experience both of these outcomes. And so before I go too deep into what happens when you have ill effects of map modifiers, I want to focus first and foremost on this idea of a B factor. So where do we get this number from? Why is it implemented almost automatically in most cryo-EM processing packages? And so this is not a new idea. And so many people have probably seen this as a Guinea plot. And so this thick red line, which is shown here, basically represents a loss of your signal amplitude as a result of your Fourier components as a resolution increases. So resolution's increasing from left to right, and this represents basically your signal fall off. And so this thick red line represents perfect signal, a perfect experiment with a very shallow fall off, which means you have great information content as far as, ultra high resolution with limited number of particles that have to be averaged. Now, as you add in noise from your data, you have these two red lines, these dashed lines that then become your traditional data, which is because the average signal is going to fall off more quickly, which means you need more data average to achieve the same resolution. And so looking at this plot, there's two main takeaways. First and foremost is that your high resolution signal falls off very rapidly, but you can measure it. And also importantly, the more noise you have, the more rapid that fall off, which is one of the main reasons why not every cryo-EM reconstruction can go to 2 angstroms resolution. So knowing this, if we have real data, so again, here's our thick red line, which basically from this complex of pyruvate dehydrogenase, it's the structure factor versus resolution. And this dashed line shown here is a fit to this fall off or the decay in your structure factors as a function of resolution. And so this fit right here is in fact what your B factor is for those data. And that's what's going to be applied to hopefully sharpen your map in a global manner. So the entirety of your volume will be experiencing this global B factor sharpening. And so anybody who's worked in CryoSpark has seen this in their refinements. And so again, it's the same thing. It's a Guinea plot. Here in green is the experimental data as a fall off versus resolution, higher resolution being here, lower being here. And the blue line's a fit, and it gives you your B factor. And so what this means is that your data will be sharpened with a B factor of 98.9 to improve your 2.6 angstrom resolution structure. Now, the pros of this, and the reason it's why it's implemented automatically in a lot of software packages, is it's simple, it's easily accessible, but the cons is that it can over or underemphasize density features in your map because it's a global modifier, which if you don't have an isotropic or homogeneous map, means it's going to differentially affect regions of your volume. And so to give you an idea, if we take a perfect image and we have image details zoomed in from A, here we have the amplitudes and we have our radial decay, right? So this image has perfect phases, perfect amplitudes. If we take the same image and we keep the phases, similar to what we have in our cryo-EM data, but we scramble the amplitudes and replace them with random noise, this leads to very poor contrast and hard to discern features. Basically, this image is barely recognizable unless you had prior information. Now, when we have a global sharpening, what we can do is we can basically measure this radial decay, this fall off, and then we can apply an amplitude correction factor to basically increase the image contrast and give more feature enhancement. And this is exactly what your global B factor is doing. It's basically applying a radial scale factor to boost or dampen the low spatial frequencies while boosting the high resolution information. Okay. Now, let's say you then have another example where these amplitudes are scaled based on your radial decay. It's a better fit to data, and it uses some sort of reference image as a base for scaling. This can lead to increased image contrast and increased feature enhancement because it's not just a simple exponential fall off. Now, best case scenario, you take the same image, same phases, but then for your amplitudes, you do a local correction where instead of having a radial fall off or a general exponential fall off, you correct locally based on your Fourier components. And this will lead to the best image contrast restoration. And so this idea is applied in cryo-EM data through what's called voxel by voxel or rolling window local scaling. And so one example is this program called LogScale, where it uses your model to generate or accurately scale the amplitudes of your structure factors to sharpen your cryo-EM map. And so what you can see is if you take this unsharpened volume and you apply this local scaling, the information content increases substantially. You can start to see sidechain information where you couldn't, and you can also begin to build the backbone much more better than previous. And so this idea of local amplitude correction or local scaling is the foundation of what some of these map modifiers like to do beyond just those global B factor sharpening. And so one of our first adventures into this was from a paper we published several years ago where we have this complex and the main catalytic component is shown here. And there's this flexible component, this docking protein that basically comes in, delivers ATP to this complex. And what we saw is that just out of a non-uniform refinement, the density for this component was much weaker than the rest of the actual complex. Even though the nominal resolution was around 2.4 angstroms, the local resolution of this more flexible component was much weaker. And so if we apply a local sharpening, we start to restore some of the features of the nucleotides in this docking protein while also recovering some more of the backbone density. But it wasn't until we applied a local mask to actually focus our refinement on this portion of the protein at the sake of the rest of it that we were able to improve the quality of density. And so what we reasoned was that it's a mixture of heterogeneity and non-isotropism in our map, as well as a mixture of just, imperfect global B factors being applied. And so through a mixture of local sharpening as well as map modifiers, we went from a density like this, which was very heterogeneous in this docking protein, to something much more isotropic like this, which allowed us to build better atomic models and interpret our data much better. And so moving forward, we started to investigate, well, given we have global B factor sharpening, local resolution filtering, as well as new programs based on either maximum likelihood or machine learning approaches, how would we best apply these to our data? And how should we best approach these given that they modify maps in very different ways? And so this is basically a synopsis of what I'll be talking about today in more detail. So in this paper, we cover all these concepts I'll cover today in more detail with all the references therein. So if I don't specifically highlight someone today, I apologize, but please, if you can, go to the manuscript where we do better due diligence to give everyone credit where it's due. So one of the first approaches we took beyond just global sharpening or local sharpening is through the Phoenix group. It's called Phoenix Resolve. And its idea is grounded in X-ray crystallography with this idea of density modification. And so what Phoenix Resolve does is it uses statistical approaches to basically calculate and model uncertainty in the cryo-EM data. And so what it does is it uses prior knowledge given what we would expect for a cryo-EM map, mostly being all of our signal is going to be at the center of the volume. And then as you move away from the center of the volume, where you have mostly just averaged noise coming from your buffer components or whatever, the signal should fall off and it should be mostly flat noise. And so what Phoenix Resolve does is it uses your half maps to estimate and correct for uncorrelated errors in the amplitudes coming in from your data. And so it takes in your input data and it calculates these and then can correct. And so this corrects errors just beyond simple sharpening. because it's not just a global application of a single B factor, but rather it's a mixture of local sharpening as well as what can come from amplitude correction within your data. And what we've seen is for almost every example in our group, protein backbone as well as side chain visibility becomes remarkably improved. But there are some assumptions in this error correction that potentially can bias interpretation, leading to sometimes overfitting or over sharpening. And so here in the example from their manuscript, you can see the original map where you have some waters, you have nice density for this map. And then as you apply Phoenix Resolve, the improvements, for example, for this phenylalanine, you can then see, holes in the aromatics, much better side chain density, much better backbone density, leading to better atomic models. And so in addition, we also wanted to see, how these different machine learning based map modifiers would also alter or improve our EM maps. And so at the time, the examples were DP Enhancer, EM Ready, as well as EM GAN. Those were the three big ones that we could get our hands on at the time. And now, of course, there are many, many others, which I'm not going to include here. But the idea with these machine learning based map modifiers is they will hopefully provide you smoother, clearer maps, decreasing the noise, improving features that are hopefully real, and improving your ability to interpret and build models into your volumes. Now, one of the things that we want to highlight and emphasize here is this has been in our group especially useful for proteins and protein complexes. And I think that, as I'll highlight later, is a result of how these algorithms were trained and built. Because the EMDB is mostly protein at this point compared to lipids or nucleic acids and those other ilk. So there's a problem though, in that in some of these machine learning algorithms, there's a black box type nature that does raise reproducibility or bias concerns, right? In particular, some machine learning algorithms are prone to hallucinations. And so how do we prevent or account for that? And then also, all machine learning algorithms are only as good as their training sets. And so if they are heavily biased towards proteins, how does that affect ligands, nucleic acids, lipids, and other non-protein biomacromolecules? And so as these are becoming widely adopted in the cryo community, we wanted to basically evaluate how these three compare to traditional approaches. And so I'm not going to go into a ton of detail for each of these algorithms because I probably won't do it the due diligence that the authors deserve. But I do want to highlight some general features of why we chose these three different machine learning algorithms. So DP Enhancer was the first that we chose because it was probably the most popular at the time. And the foundational idea of DP Enhancer was this local scaling that I discussed earlier in the talk could be applied to basically any EM volume if you have an atomic model. And so the idea is this low scale takes in your input model, takes in your raw map, and uses that as a way to scale your amplitudes locally based on your model and input map. DP Enhancer tried to circumvent this by not requiring an atomic model, but instead learning behaviors, excuse me, different noises and amplitude scaling within the maps. So you can go from raw map or half maps directly through DP Enhancer using their end-to-end UNet architecture, which is really common in biomedical imaging techniques, as well as then to output a masked, locally scaled processed map. And so they provided multiple different models within their algorithm to apply this, depending on whether you wanted high res, medium res, or a mixture. And so that way you can then improve the quality of your EM map without requiring an atomic model. And so in their example, you have an integral membrane protein where you have this, micelle density, which is very noisy. And then here's the actual raw EM data. In the published structure from the authors, you can see that you can visualize backbone, alpha helices, and good density, but there are regions that are over sharpened and weaker because of global sharpening that's been applied to this volume. Using DP Enhancer, all of the features in this map become much more robust, loops are recovered, and it allows for a much better model to be built. And so, as a proof of principle, this was a great way to segue into map modifiers. Now, another approach that doesn't use a traditional end-to-end UNet like Deep Enhancer is EM-GAN, which uses this generative adversarial network. And the idea here is that unlike a purely deterministic architecture, it uses this adversarial setup where it has a generator and a discriminator to where it tries to make your EM map more interpretable, while at the same time using a discriminator to help basically ground the improvements so that way you don't overfit or hallucinate within your EM data. And so they gave three examples of varying resolution, where in each case, what you can see is the backbone connectivity and the interpretability improve for each one. And so, because these are working in the 3 to 4 angstrom range, this is really good to where in the atomic model building regime, it's very difficult sometimes where you lose connectivity, you lose sidechain density. And so, this is a great way to improve general connectivity across the EM volume. Another machine learning algorithm we investigated was this EM Ready, which uses a Swin transformer or a shifted window attention. And so this is different than the other two, then that it tries to basically maintain connectivity and also maintain some sort of knowledge about disparate regions in the EM volume rather than just focusing on local regions. And so what this does is it can capture both local detail within the patches that it's using, while also maintaining a global context or these long distance relationships across the map. And so this is really key for lower resolution volumes or volumes where you have a large range of local resolution within your EM volume that'll help maintain this connectivity and this chemical bonding. And so as kind of summarized here, I'm not going to belabor the points here, but like each of these machine learning algorithms use a different core architecture. They use a different learning strategy, both in how they input their EM volumes and how they modify the EM volumes. And as a result, they have very different strengths, pros and cons for each one. And so there are specific use cases for each one. Like I said earlier, not one of these machine learning algorithms is going to work on every single EM volume. I think that's unfair to the authors because, every volume is wildly different, as you would imagine. But in the end, what we have to do is design best use case scenarios for each one because they could hallucinate, or in some cases, as I'll discuss, they'll erase subtle or even non-protein densities. And so, how do we evaluate this? As you saw earlier, there's a lot of different ways to examine how well your model and your map are behaving. Obviously, if you have proteins, you can use Ramachandran, Rotamer outliers, or Kablam, Molprobity. You can also use z-scores or map to model fit. There's also q-scores, which offer a per atom correlation. But regardless of which value you're investigating or monitoring, ideally, anytime you have a map modifier, your metrics should improve. If they don't improve, then something has happened. And so these should always be used in a complement to visual inspection. Don't just run these as a black box and take everything as gospel truth at the end of the day. And so we elected to basically take various input volumes, run them through traditional map sharpening algorithms as well as these machine learning algorithms, and then evaluate what happens on the other side. And so we selected to use Phoenix Resolve, DP Enhancer, EM Ready, EM GAN, which is a mixture of maximum likelihood density modifiers or machine learning based map modifiers. We would update each atomic model into our map using Phoenix Real Space Refine. And then just very quickly, we would use a Q-score analysis to evaluate protein backbone or side chains or our ligands in our volumes. Our test cases were 7 different proteins ranging from molecular weight and different sizes, different resolutions, different microscopes, as well as different compositions. So T20S proteasomes, just purely protein. Nucleosomes are, DNA plus protein. Connexins are an integral membrane protein with annular lipids, as well as different types of ligands across these data sets. And so by simply using Q-score, we elected to use this because A, it's easily quantifiable. It has a very easy way to identify what's a good value versus a bad value. Ideally, the closer to 1, the better the fit of the model into the density. The closer to 0, the worse the fit. But importantly for us, it was built directly into Chimera X. So we could load in our volume, we could load in and just quickly assess how things were improving with or without the map modifiers. And then we could do a per residue per amino acid type calculation. And so for our first example, we took the T20S proteasome, which we've been studying by cryoEM for over a decade now because it's a very good benchmark protein. There's no ligands to be known with this current T20S proteasome. In the deposited map, this went to about 2.4, 2.3 angstroms resolution. So you can see backbone density, you can see side chain density. When we apply Phoenix Resolve, the backbone density improves substantially. Whereas if we apply DP Enhancer, we also got general improvements. And if we look at this for just basic Q-score backbone quantification, we see compared to the deposited map, which is shown here in this BioM plot, Phoenix Resolve, EM Ready, and EM GAN had the most improvements for the protein backbone fits across all the volumes tested, while some of the other different machine learning algorithms had a bimodal distribution, depending on the quality of the input map and the different features. And so to give you an idea of what this looks like, so in our deposited map, you can clearly place this alpha helix. You can see where the carbonyls will be oriented. In our Phoenix Resolve, the improvements in our density are marked, where we can clearly see the carbonyls for our backbone are much better placed. And so this, as a result, leads to better refinement statistics, better model building, better placement of our rotamers. And even in Deep Beam Enhancer with their higher res volume model, you can also see improvements in the placement of our carbonyls. Now, when you compare protein side chains, the quality gets much more different across the different maps and across the different algorithms. So in general, again, Phoenix Resolve and EM GAN do quite well in improvements, but you do see that across these different packages, they have very wide behaviors depending on the type of the input volume. And so because of this, you have to be careful with how things are improved. For our deposit map, you can clearly see that this is an arginine. The guanidino group can be placed quite well. The Phoenix Resolve density is markedly improved, as well as the DP enhancer density. Now, this is great for placing your side chains, but one thing that we did observe is that in the 20S proteasome, there is one tyrosine that experiences an alternate rotameric conformation. That's been observed in several EM volumes. In the deposited map, it's basically a 65-35 split in how it's being refined. Phoenix Resolve improves this to almost a 50-50. This is retained within the map that's been modified. But in DP Enhancer and the other map modifiers, it basically collapses to a single rotameric position, which in some cases is not the end of the world. But for an example of, an enzyme or something where this alternate rotamer is critical for catalytic activity, this could be detrimental to any type of foundational understanding. And so one of the things that we quickly took away from our analyses is that in general, almost every map modifier we tested improved our carbonyl and our side chain density. Phoenix Resolve in general was the best performer with the most reproducible and consistent improvements. For example, our beta strands and helices became much easier to trace. Our carbonyls were better. But one of the dangers that we observe from this is that all these methods, except for Phoenix Resolve, tends to prefer a dominant side chain conformation. And so as a result, diversity can be lost depending on the context and the quality of the EM data. Now, moving into some very specific test case studies. Here we have a nucleosome. And so this is a mixture of protein and nucleic acids, 2.6 angstrom resolutions nominal, which overall is pretty good. In the deposited map, what you can see is for the bases, you have clear connectivity for traditional Watson-Crick style, but then on the backbone of the nucleic acid from the DNA, it's much weaker at the periphery away from the center of the volume, even though it has fairly good Q scores. Now, Phoenix Resolve had mixed results where the bases were improved. The backbone of the nucleic acid that's adjacent to the protein was quite improved compared to the reference deposited map, but at the periphery, things got quite worse for some reason. DP enhancer, they had better base separation, but then the backbone wasn't as improved. And so this could lead to better interpretability of what an individual base was. EM Ready had improved connectivity in the EM backbone, I mean the nucleic acid backbone, not much improvements in the bases, whereas EM GAN had mixed results, improving the backbone while also having base separation. And so the takeaway from here is that sometimes the bases, the quality of the density was eroded, became less interpretable, sometimes it was improved. Q scores in general were modestly improved, if at all, but in general, EM Ready appeared to do the best with nucleic acid backbone clarity. And so, one of the things that we worried about from this analysis is that because these machine learning algorithms are overly protein centric, they may be biased towards nucleic acid backbones and away from general nucleic acid features. And so, anyone working with nucleic acid protein complexes should take this into consideration. Now, for alcohol dehydrogenase, this is a 2.9 angstrom structure. It's much smaller than the nucleosome. And so in the deposited map, it has a very nice density for this NAD cofactor, as well as this catalytic zinc within the active site. Phoenix Resolve had a much improved density for the NAD cofactor, as well as some of the surrounding residues, but the zinc surprisingly looks different than the deposited map. For DP enhancer, there was a mixture of improvements where the zinc is somewhat retained in this catalytic site, but in general, the best improvements were seen for the NAD cofactor, but overall things kind of decrease as far as Q score. For EM Ready, even though the NAD doesn't fare as well compared to the others, the zinc ligand is surprisingly missing in this modified map. Whereas in EM GAN, you get improvements in both the NAD cofactor as well as the coordination of the zinc in its local environment. And so what's interesting is because these are proximal in their local chemical environment, they're experiencing different types of map modification. And so in general, as we kind of expected in hindsight, the NAD cofactor fares quite well. It resembles a protein backbone. It's easily recognizable. It's kind of a long, tubular type density. Most of the Q scores were improved, especially for the NAD cofactor, but in general, not great improvements. But the catalytic zinc metal experienced very mixed results. In some cases, it's completely lost. And so the risk here is that it's inconsistent depending on where you're looking. And so in this case, which one of these maps that have been modified would you use for your interpretation? Is the zinc there or not? Now, beta-galactosidase has been a test specimen in cryoEM for many, many years now, and this 2.2 angstrom structure of beta-gal with this PETG ligand has been used in numerous examples. And so in the deposited map, you can see a magnesium as well as coordinated water molecules with really good density, which you would expect for a 2.2 angstrom map. And there's also the sodium that's located within this active site and this PETG ligand, which surprisingly does not have great density for this benzyl group. Now, in Phoenix Resolve, surprisingly, the magnesium and the waters have much more spherical type density, which is going to allow for their individual coordinates to be better refined, better placed. The sodium does fairly well, but the PETG ligand does quite worse compared to the deposited map, which would, without prior knowledge, you wouldn't really be able to tell that this is in fact a PETG ligand. DP enhancer, you see that the magnesium and its water coordination is now blurred to where you don't resolve individual waters, but instead you see kind of this tetrahedral, octahedral style density. And then the sodium is still retained. Whereas in EM ready, you see loss of the coordinated waters around the magnesium, and the sodium is completely gone for some reason. Whereas, the PET ligands mostly retain compared to the raw map, but surprisingly differential effects on the water. Now, the one thing that's also interesting is in EM GAN, you maintain the waters quite well as the sodium placement, but the overall quality of the sodium density does decrease a little bit compared to the other maps. And so what we saw here was there's quite mixed results for the metals and the waters, which is surprising given that the scattering coefficients of sodium and water are not all that different. The Q scores overall are either similar or worse for many of these, but Resolve and EM GAN do seem to behave the best compared to the other two algorithms. Now, of note, the PETG density is surprisingly weak across all of these maps, whether it's just a global B factor sharpening or whether it's Phoenix Resolve or the machine learning. And so one of the things that's risky here is that the metal coordination as well as the ligand density are quite unpredictable given just prior information. Now, this is going to hit probably close to home to Steve, but this is his connection structure. It's a 1.9 angstrom structure, very high quality density with, it's an integral membrane protein, which has two belts of annular lipids. And one of the things that we saw was if you're looking down the central axis of the protein, these are the annular lipids that are shown here. And in the deposited map, you can see quite good placement of each of these annular lipids all the way out to like a third belt, so to speak. In Phoenix Resolve, you get great enhancement of the protein backbone. The carbonyls really pop and the waters shine in this map, but you start to see some of the fall off in these outer lipids for some reason. Now, in DP enhancer, you get good backbone connectivity, but you lose resolution overall compared to the raw map because it's emphasizing connectivity versus some of the high res features. But these distal lipids are completely lost in this map that's been modified. Now, in EM Ready, what's surprising here is that only two of the annular lipids that have the highest occupancy are retained. All the other ones are completely lost. In fact, these outer two, there's almost no density, so to speak, for these two lipids. And then in EM GAN, we have a similar phenomenon where we have the first belt is more or less retained, but then the outer ones are completely lost. And so if you have a protein where these annular lipids are important for function, only in one of these modified maps would you really be able to build this network of lipids. And so in general, what we saw is that near the protein, things are enhanced or retained. Whereas as you move away from the center of the protein into the lipid belt, those densities are either suppressed or completely removed. And so as I worry about with some of these, high res integral membrane protein structures, the interactions between these annular lipids and the protein could be completely subverted depending on the map modifier. And so again, this highlights the importance of comparing both the raw, the global sharpened, and these modified maps, because if you take just one as the gospel truth, you may fall into a pit of misinterpretation. So despite similar features, despite the fact that these annular lipids look like what you would expect for, a connectivity of the protein backbone, there is a concern that the protein-centric training sets biased against these long tubular annular lipid type features. And so there might be interest in actually training some of these map modifiers on integral membrane protein-centric data sets. Now, this I would say is probably one of the most concerning that we evaluated. And so this is a NAV channel, NAV 1.7. Nominal resolution was 2.7 angstroms, and this is bound to the small molecule called bupivacaine. And so you can see in the deposited map, you have clear placement of this very asymmetric molecule where you have this 6-carbon ring placed here, and you can see how it fits quite well into the EM density. And then also the local chemical environment is also very, very well resolved. And so what was surprising in the Phoenix Resolve density modified map, you get improvements in the backbone of the surrounding residues, but then the density for this bupivacaine changed quite a bit. And so one of the things that we reasoned is that this may actually be a mixed pose binder, or it could be mixed occupancy, and that in the Phoenix Resolve with the better amplitudes, you could start to understand how this might bind into potential different poses. Now, things get really wonky in the machine learning algorithms where in DP Enhancer, EM Ready, and EM GAN, all three almost completely scrub the maps of this bupivacaine density. And so the concern here, is this a result of training bias? Is this a result of, weak EM density in the raw half maps? Or is there something else that's going on here that needs to be further investigated? But, a cautionary tale is if you went from this straight into DP Enhancer, EM Ready, or EM GAN looking for your ligand, you wouldn't see it. And so there is a very careful tale here that, again, you want to look at the raw, global sharpened, as well as these different modifiers before you come to a conclusion, right? So in general, again, the protein density was actually handled quite well across all of these different map modifiers. However, only Resolve was able to maintain the ligand density. Now, is this a result of it keeping the phases and modifying the amplitudes? Is it a result of something happening in the machine learning algorithms? We don't know for sure, but it also emphasizes that there may be different poses or other information in the raw map that could lead to more conclusions. And so, despite improvements in the protein itself, the ligand density is completely gone in these MO-based maps. And so, again, which one of these do you trust, right? Of the 5 maps I present here, which one do you actually trust? And I'll leave you, to take that into consideration later on. Now, another thing that we also observed is looking at hemoglobin. So this is a 2.4 angstrom structure of oxyhemoglobin, 64 kilodaltons. So it was the smallest that we evaluated in our test set. And so in the deposited map, you can see clearly the heme cofactor. You can see the vinyl groups and the propionates have good density. You can see the histidine and coordinating distal and proximal ligands, as well as a bound oxygen at the heme iron. In our Phoenix Resolve map, you can see that the vinyl groups on the heme are improved quite a bit. And the O2 density, the oxygen density is retained, but it's not as strong as in the deposited map. And we start to see some difference in the connectivity between this oxygen and its iron that it's bound to. Now, in DP Enhancer and EM Ready, the oxygen density is almost completely gone in both of these. And what's also intriguing is in EM Ready, the connecting proximal histidine to iron coordination density is more or less gone compared to the other 3 maps that we observed. Now, EM GAN, similar to Phoenix Resolve and the deposited map, has good oxygen density, but it loses the connectivity to the heme iron, and then it maintains it to the proximal histidine ligand while improving the quality of the heme density. And so what we saw again is the protein density behaved quite well in these tests. Not surprising given the bias in the training sets for all these different algorithms. whereas only Resolve and EM GAN maintained the oxygen ligand density. The heme vinyl groups were better resolved, but there's differences in how the heme iron was either connected to the oxygen, it was either lost or weakened. But in general, the heme density quality was varied across the maps, but general was retained, which we imagine is because it's a big cofactor, right? Now, what's interesting is in the DB enhancer and EM ready maps, the heme density was much smoother than what was deposited in the raw maps. And we think this is a result of how these map modifiers are quote unquote interpreting what that density should be. And so again, the heme and oxygen densities, the ligand were different between the map modifiers. And so which one do you trust? Do you trust, one of the other? Do you trust the deposited map only? So be careful in how you interpret these data. Now, in general, what we saw for all the machine learning methods is that ligands, waters, and metals were either removed or degraded in general. Some cases they were improved, but overall there was a considerable loss in density for these compared to their proteinaceous counterparts. And so you can see here, looking at Q scores of the various ligands, that in the deposited structures, they actually behaved quite well, and there wasn't a huge disparity in the quality of the Q scores. Phoenix Resolve, things started to stretch out quite a bit, which could be a mixture of, not updating the refinements properly or also, how the things were modified. But then when you get into the machine learning algorithms, you start to see marked decreases in overall ligand quality as a result of these Q scores either reaching zero or going below zero, which means effectively there's no density for those ligands. And so overall, that's quite concerning in my book. In fact, again, highlighting this bupivacaine example, in the machine learning maps, that density was, you know, completely gone. There was no indication that there was any ligand bound. And so there is a risk here in that if you only use one of these map modifiers, you could have a false negative ligand interpretation if you're doing drug discovery. And so in a high throughput regime, if you're only using one, you could completely skip over something that in fact actually does have your ligand of interest bound. And so in the last couple slides before I give away to questions, what I really want to highlight is that in almost all cases we evaluated, most of these map modifiers, whether it's global sharpening, local sharpening, resolve, or the machine learning algorithms, they improved the backbone connectivity and the backbone density quality, with resolve in general having the best overall improvements. Sidechain interpretability was also improved in general across the board, with one caveat being that heterogeneity is typically going to be dampened in these different map modifiers. Now, for larger ligands, which include the NAD cofactor, the heme, and kind of PETG, they were retained or improved much better across the board. Bupivacaine was the one that is the outlier here, even though it's large compared to most ligands. Now, smaller ligands, this includes metals, waters, and their coordination environment, in general, were lost or weakened, or were just highly variable compared to the surrounding improvements that we observed. And then nucleic acids and annular lipids, they were the most variable depending on the software package. And so there is a selective retention loss depending on the density class. And so depending on whether you're looking at lipids, nucleic acids, different types of ligands, you could have quite different outcomes depending on which map modifier you use and the quality of the incoming input EM density. And so how can you responsibly use machine learning map modification? Well, the strengths are clear, right? Protein density, improved model fielding, interpretability, at least for proteins in general, it's across the board an improver. Most algorithms are heavily biased towards proteins, not too surprising given what's in the EMDB, but this can lead to erasure of ligands, right? Especially if they don't have continuous density that kind of quote unquote resembles what you would expect from a protein. And so Is this directly a result of heavily biased training sets? Is there something else that's going on that we need to further evaluate? I'll leave up to the authors. But the unpredictable nature of metal ion lipid modification and ligand modification means you should never rely on just one single modified map alone, especially if it's ambiguous, low occupancy, or heterogeneous. Now, I would recommend comparing your global B factor sharpened or your locally filtered with your modified maps to build your narrative. And so again, you have to quantify this, right? Whether you're using Q score, real space, Z scores, map to model scores, whatever it may be, you have to analyze your qualitative outcomes and quantify them so that way you have faith in what you're putting forth in the public domain. Because you don't want to claim that you have an oxyhemoglobin structure when you have no density for the oxygen. Now, practical recommendations. One thing I would say for everyday users is, again, compare across maps. There's no harm in comparing across maps, right? But again, you need to validate what you're building, ligands, nucleic acids, whatever it may be. And at the end of the day, be explicit and be transparent. Document everything. Whatever map you use for model building, refinement, interpretation, it needs to be documented. It needs to be in the public domain, right? The EMDB is a free repository. So if it went into your manuscript, put it into the public domain, right? Machine learning tools are complements. I would not say they're replacements as of yet from the global sharpeners. And so you need to treat them in some caution. And so again, I advocate for critical thinking. Don't just blindly trust anything that's put out there. And so every map, again, every map you use for interpretation, put it out into public domain. There's no harm because at the end of the day, you don't want to get an email from someone saying, hey, you claim this, it's not in your map, put your map up into the public domain, especially if it's on CPEM or the different bulletin boards. So in general, to summarize, balanced use is the best approach, right? Cryo-EM is transformative, but you have to be careful in how you interpret your maps. Map modification is necessary, whether it's global or local, but the method and your interpretation matters. And so protein density, you're probably going to be in pretty good shape. Beyond that is when you get into the danger zone. Now, in your publication, consider, would you publish a ligand model if it only appeared in a machine learning modified map and it wasn't present in any other maps? Which one do you trust? If you have two map modifiers that disagree on sidechain orientation, which one would you trust? Which one would you publish and why? And then importantly, again, which ones do you deposit in the EMDB? I advocate for everything, but I'll leave that up to you, it's your own volition. And then which resolution ranges do you use the different programs? One thing to emphasize here is that if you have a 2 angstrom map, you probably don't need most of these map modifiers except for maybe Phoenix Resolve. But if you're in the 3.5 to 5 angstrom range, you can probably use more than one. And so with that, I want to thank my group, especially Brian Cook and Ray Berkeley, who were the lead authors in the study, and the rest of the group in funding for considerations and discussion.
Mark, thank you very much. That was a great talk and really a diligent effort in helping the field understand the strengths and weaknesses of some of these tools. It can be super eye-catching what these programs put out, but you have to be very critical, for sure. So I'll try to address to the best of my ability a couple of questions, but Omar, I think we're kind of tight on time, so I'll just try to do a couple. So I thought a good question was, have you had any experience or do you have any insight on assessing whether an AI enhanced map should be well trusted before building into it? Is there a way to kind of get an assessment of, hallucination or not before you spend all the time building your model into it?
That's a great question. So one of the things that we have seen is right out the gate, if you open up a map that's been modified with one of these machine learning algorithms and the default thresholding in Chimera, if you see weird artifacts like grid latticing or anything that pops out in a non, you know, component region of a complex, that's usually an indication that it grasped onto something that you don't really want. Whether it's, a neighboring particle, a masking artifact, or something. And usually, once we see that, it has highly variable results. And so that's the first thing that we acknowledge right out the gate is, what does it look like when you first open it in Chimera? Or, if you're running DP Enhancer or something through CryoSpark, then what does the Z slice look like? Like, do you see these weird features that are popping up? If so, you probably shouldn't trust it or be very careful in how you interpret it.
Yeah, great. Thanks. Um, can we do one more, Omar?
Yeah.
So it seemed that you emphasize that non-protein component. I'm trying to generalize some of the people's questions. So non-protein components weren't handled as well as protein components. I thought you did a good job kind of summarizing, but there's lots of questions about this. Did you see any kind of correlation with, or I guess, did you have a chance to look at anything with glycans and maybe more in general, any correlation with like prevalence of these, I'll just call them all ligands in the EMDB or PDB versus how well they performed with these tools?
That's a great question. So we initially tried to find a really good example where the glycans were modeled well. But I think in general, what we found was that because of the inherent heterogeneity of the glycans themselves, it made things really wonky. And so we wanted to be very careful in how we interpreted that because, you know, there is inherent heterogeneity both in what glycans are attached sometimes and how they position in 3D space. And so we didn't want to pick and choose one example and then, make these broad spectrum comparisons. But I would say in general, they kind of fall into the annular lipid example, where the further away you get from the protein, there's a very rapid fall off in quality and how they get interpreted.
Cool, thanks. I might invite Mark if you have time to maybe peruse some of the Q&A
Sure.
during the next speaker so we can get them up and running on time. And thank you, Mark, again for a great talk.
Absolutely. Thank you.
All right.